Nuestra siguiente ponente es la doctora Sonia Sultan. Ella nos va a estar presentando en inglés, pero si ustedes tienen preguntas, bueno, las pueden hacer en español, no hay ningún problema. Estaremos haciéndoselas llegar a ella en inglés. Sonia Sultan nos va a presentar la ponencia Organism-Environment Co-Construction, a Case Study in Plants. La doctora Sonia Sultan es profesora en Biología y Estudios Ambientales en la Wesleyan University. Ella tiene estudios en Historia y Filosofía de la Ciencia en Princeton y un doctorado en Biología Organísmica y Evolutiva en Harvard. Y su grupo ha estado trabajando desde hace varios años en torno a los estudios de plasticidad en el desarrollo en plantas, y ha trabajado tanto con la integración teórica de la plasticidad en la biología evolutiva, como con aspectos que tienen que ver con una de las ideas que también desarrolló Lewontin, que es el tema del rol o el papel de la plasticidad fenotípica en el proceso evolutivo. Ha publicado muchísimos trabajos, en particular hay un par de libros que son muy importantes, ¿no? Uno que es muy importante, Organismo y Ambiente, Ecología, Desarrollo, Construcción de Nicho y Adaptación, que es un texto bastante amplio con mucha evidencia sobre este campo. Entonces, sin más, le damos la palabra a la doctora Sonia Sultan. We appreciate very much having you in this round tables around the works of Richard Lewontin, dear Sonia. So thank you very much for accompanying us here.
Well, thank you very much, Lev, for the invitation to participate. My Spanish is extremely imperfect, but I was able to learn a great deal from both of those presentations, so thank you for them as well. It's really an honor to be part of this event, recognizing the legacy of Richard Lewontin, whose ideas continue to so profoundly influence our thinking as scientists and our thinking about science. I'm going to give a third perspective, in addition to the two quite different ones we've already heard, and talk about one particular focus of Lewontin's work, the relationship between the organism and its environment, and what he described so beautifully and subversively as the triple helix between genes, organisms, and environments. And along the way, I'll present some findings from my group's research on plant ecological development that will illuminate this organism-environment relationship. So some of the themes are those that were already mentioned by Ali, but you'll see them, I think, a little differently when there's data next to them as well. So let me make sure I can advance there. So as you're all aware, for decades, the dominant approach in biology has been a gene-based one, in which an organism's DNA is understood to provide specific developmental instructions for all of the traits of that organism, whether it's a bacterium or a plant or a frog, or indeed a human being. So according to this is the genocentrismo that Ali mentioned. So according to this view, an organism's set of genes, its genotype, specifies that organism's traits and behavior, its phenotype. And we can know pretty much everything important about an organism by knowing its DNA sequence. So this purely internal genetic information is our primary focus in this conventional view for understanding the causes of development and variation. At the same time, the organism's external environment dictates its function and hence its reproductive success to filter genetic variance in the process of natural selection. And as a result of those principles, we can study adaptation simply as change in allele frequencies. So this is the modern synthesis view. And for biologists, this framework has led for decades to a research focus on genetic variation per se, and a strict separation between the internally determined organism and its externally defined environment. As a result, a standard experimental design for studying variation. Now, can I ask you, can you see my cursor? Yes, super, okay. In a standard experimental design, we would place genetically different individuals, so genotype A and genotype B, for instance, in a single control environment that's meant to be developmentally neutral, and therefore to to reveal the genetic programs of these different organisms. In this kind of experiment, since all the organisms develop in the same fixed environment, each genotype produces a single characteristic phenotype, and any differences between them must be due to differences in their genes. So the results of this kind of control environment study will always confirm a gene-based model. So this is the prevailing approach, and in contrast with this approach, Lewontin argued for biology to reintegrate the organism and its environmental context. And to do this, he advocated a different way of studying organisms that brings environmental variation into the experiments. And that's the approach we use in my lab and many other labs that study what's now called ecological development, that is, development in environmental context. So the general approach for this kind of work is to make several replicates of each genotype of interest. So here I'm showing cloned replicate plants of genotype A and of genotype B, and then grow those replicates in several different ecologically realistic environments. And then we can examine the results by plotting the phenotype produced by each genotype in each environment for any trait of interest. So this can be a body, the size of a body part, or a metabolic rate, like photosynthesis rate in a plant, or reproductive output, or any trait. And then each genotype's pattern of phenotypic responses, this whole pattern that characterizes the genotype, is called its norm of reaction. And those response norms or patterns can be compared for different genotypes. So this kind of data gives us a very different picture. First of all, norm of reaction data makes explicit the point that the phenotype produced by a genotype depends on the environment. In other words, the genotype is a repertoire of contingent responses, not a self-contained script for a single determined outcome. So this approach lets us study the organism's traits as the joint result of both genotype and environment. As Lewontin emphasized, this model corrects the causal asymmetry of a gene-based model. And now, of course, we understand that at the molecular level, genes are differently expressed in different environmental conditions because environmental factors enter into cellular and nuclear regulatory processes. So now we see why norms of reaction exist. And just as the effect of the genotype on the phenotype depends on the environment, the phenotypic effect of the environment depends on the genotype. Since genotypes can differ in perception of the environment and response to the environment due to variations from one genotype to another in these regulatory interactions at the molecular level. So today, I'm going to very briefly present some results of EcoDiva work in my lab with these very common herbaceous plants to examine how the organism and its environment define and construct each other. And I hope that gives you some ideas for the discussion later on. So in our experiments, we collect seeds from natural populations of Polygonum, and then we clone or inbreed each genotype collected from the field and raise genetically identical individuals in different ecologically relevant environments in the greenhouse. So for example, because light intensity varies among and within plant habitats, we use shade cloth and green filters to create light environments in the greenhouse, and then ask whether polygonum genotypes express different phenotypes in those different environments. In a case like this, I'll show you a lot of data for these light and shade experiments. In these cases, it's particularly interesting to study leaf traits, because leaves are the organs plants use to collect light, which is what plants eat. So leaf traits have particularly important functional and fitness consequences. Okay, I'll show you some data. So this is a plot showing the norms of reaction for the size of polygonum leaves produced in two different light environments in the greenhouse, low and high light. Each line, is one genotype that was cloned and grown in both environments. And this experiment had 8 polygonum genotypes. So notice a couple of things. First of all, notice that all the genotypes made different leaf phenotypes in the two environments. So, for example, we can't ask the question, what is the leaf size of this genotype? Because it depends on the environment. But similarly, we cannot ask what leaf size is produced in this environment, because it depends on the genotype in question. And that's the causal symmetry that Lewontin draws our attention to. So this is what the leaves actually look like. Leaves produced by plants growing in high light are narrow, which is functionally a good idea since they don't lose too much water from their surface area. Plants grown in low light produce these very large, sort of broad leaves that increase the plant's ability to catch photons in low light, where photon flux density is much lower. So keep in mind, these very different leaf phenotypes are produced by the same genotype when it's growing in different environments. Leaves produced in contrasting light environments are also anatomically quite different from each other, although you can't see that in the photo. Plant leaves produced in low light spread the tissue out much more thinly, so they're very thin, and that increases the surface area for collecting light per gram of tissue. And in contrast, leaves produced in high light are quite thick, with multiple layers of light processing structures that let those leaves utilize the very high photon flux density available to them. I want to add one more bit of data to the norms of reaction for leaf size. So here I'm showing you a third, more extreme low light treatment to add an evolutionary observation. So if we look now on this in large scale for leaf size, we can see that the polygonum genotypes converge on more or less similar leaf sizes at 100% light, at full light, and at moderate light. But at this very low light level, a subset of the genotypes produce extremely small leaves, possibly because for these genotypes, some kind of maladaptive threshold has been crossed. So these data illustrate the fact that genotypes can produce similar phenotypes in some environments and quite different phenotypes in other environments. Also, a genotype can produce a relatively high trait value in one environment, but a low trait value relative to others in another environment. So these data illustrate the point that for a given set of genotypes, the variation that's available to selection is not determined only by what genotypes are present. Instead, the genetic variance is co-determined by the distribution of different environments. And that's, I think, a very counterintuitive point, a very important one for evolutionary biologists, because it means that the phenotypic responses of organisms to their environments shapes their evolutionary potential. Okay, so back to low and high light, plants grow in different light environments also change their body proportions very dramatically. So here you can see that in highlight, plants invest about 15% of their body mass in leaves. That increases, that allocation to leaf tissue doubles or triples as light is increasingly reduced. And then in other experiments we've done, of course, a lot of them, in other experiments we've found that these same genotypes also change their body proportions in response to different soil conditions. So for instance, when growing in soil that has little water or low nutrients, plants invest a higher proportion of their body mass in roots, and making a larger, more extensive root system allows them to more effectively collect those limited soil resources. We've also done some work showing that more subtle aspects of root development, such as the form of roots and their location, their placement in the soil, can also change in response to the environment. So here I'm showing you quite an extreme environment for plants to grow in, flooded soil. I think you can see the water is actually just above the soil surface initially. And this is a very stressful environment for plants because plant roots need to collect oxygen. But in flooded soil, the air spaces in the soil are instead filled with water. So polygonum plants, when grown in flooded soil, produce this very surprising and unique kind of root system, which I hope you can see here. Very, very fine hair-like roots produced on the surface of the soil, that is at the interface between the soil and the air. So that's a really interesting response because it allows the roots to directly take in oxygen from the air, which is a part of the environment that roots don't normally contact at all. And it creates a supply, a plentiful supply of oxygen from the air above the soil. Okay, so to summarize, a single plant genotype will produce very different phenotypes depending on its environment. So notice that these changes in response to the environment are not minor tweaks to a predetermined developmental program in the genes. This isn't noise, tiny noise around some kind of genetic signal. These are significant formative changes to essential functional traits. So that's one thing. These are big adjustments. They're meaningful in terms of phenotype. And also these environmental responses are very specific adjustments. They work in response to particular environmental signals to modify the phenotype in ways that increase the availability of whatever environmental resource is most limited. So environmental conditions provide specific information that elicits specific and often functionally adaptive developmental outcomes. So by participating in developmental processes, the environment shapes the organism. And then these developmental responses in turn shape the external environmental conditions for the organism and other organisms in its habitat. So for example, the plants grown in low light, this is a different related species, plants grown in low light that produce larger leaves intercept a higher percent of incident light and therefore cast more shade on their neighbors. These thinner leaves also decompose more rapidly in the soil, so they increase mineral cycling rates in the soil. For the surface roots produced in flooded soil that I showed you, those roots bring oxygen into the soil that other organisms can utilize. So the organism's developmental responses change the external environment, and they also profoundly change the environment that the organism itself experiences. So for example, plants grown in low light that increase the surface area of their leaves experience an environment in which more photons are available. Plants in flooded soil that produce surface roots experience an environment in which plenty of oxygen is available. And plants in dry soil that build larger root systems inhabit soil where more water is accessible. Animals also express this kind of functionally adaptive, specific responses to environmental conditions, of course. Here are a couple of very charming cases. When tadpoles of the brown frog develop in the presence of a salamander predator, they develop this very puffy, enlarged phenotype that's too big for the predator to eat. These sea urchin larvae develop differently. When they develop in water where food is scarce, they produce these larger feeding structures to collect food. And these developmental responses change the external environment of the animals. So when tadpoles respond in this way, their salamander predators will eat more of something else. By sea urchin larvae that develop in these larger arms will eat more of what they're trying to eat, and other organisms in their habitats will have to modify accordingly. So they change the environment the organisms experience, because here as well, because these sea urchin larvae create an environment in which more nutrients are available to them. And these tadpoles create an environment that's predator-free. So organisms don't just respond to their environments by developmental plasticity, they change their external environments, and they change their own experience of those environments. And here is where, as Lewontin argued, the organism, the actual realized phenotype, the organism and the environment are one. So, in other words, we've moved beyond an internally determined organism that confronts an externally defined environment. We've moved beyond the notion that the organism and its environment are separate entities that interact each other, to the point where we can see that they co-construct each other. In other words, environmental conditions influence development, so the environment shapes the organism, and that environmentally influenced phenotype in turn determines the organism's experience of its environment and its impact on external conditions. So the last thing I'll show you is just to very quickly return to Polygonum to add one more dimension of eco-devo data. Recently, we've been conducting transgenerational eco-devo or norm of reaction experiments, where we grow replicate parent plants of each Polygonum genotype in contrasting environments, such as shade versus sun. And then we collect their seeds and grow the seeds to compare their offspring. And the goal is to test if a plant's development is influenced by the environment its parents grew in. So in these, the way we design these experiments, the only difference between these two sets of seedlings, for example, those produced by a shade parent and those produced by a sun parent, they're all the same genotype. So the only difference is the environment of the parent. And then we can compare the effects of those alternative parental environments in different growth environments for the seedlings. I hope that's not too confusing. They're complicated. Okay, so this plot shows some of the results of one of these experiments. So here I'm showing you again leaf area, and this is SLA, which is specific leaf area, or the leaf area per gram, so the thinness of the leaves. The dotted line shows the seedling progeny of shaded parents. The orange line shows the offspring of parents that had been grown in the sun. And this shows the two different, the offspring developing in shade and offspring developing in sun. So as you can, and I've shown that I've pooled all 5 of the genotypes that were in this experiment in each of these collective norms of reaction. Okay. So offspring of shaded parents, offspring produce larger and thinner leaves when their parents grew in the shade. But that's only true when the offspring themselves are growing in the shade. Because when the offspring are growing in the sun, they don't express that effect of the parental environment. And these, again, these are functionally and ecologically very important traits, and they show quite a big impact of the parental environment, but only in some growth conditions. Okay, so parental environments can influence the offspring's environmental responses, their norms of reaction to sun versus shade, for traits that are important to function, competitive interactions, and fitness. We found in our experiments that these effects, these inherited environmental effects, can persist from the grandparent generation. That's as far as we've gone, although I think we should keep going and see if they can persist even longer. Okay, so these data and transgenerational data from many other labs show that organisms can inherit environmental as well as genetic information. In Polygonum, our further work showed that these inherited environmental effects are mediated epigenetically, as they are in many plant and animal systems. And in Polygonum, in this case, the inherited effects of shade and sun, and also of dry versus moist soil, are mediated by DNA methylation changes that persist, again, for at least 2 generations. So these molecular epigenetic mechanisms allow environmental signals to be internalized into the genome itself, and therefore into the process of heredity for potentially several or even many generations, depending on the system. So, you know, for me, this is a really interesting result because it means that the genomic information that we consider internal to the organism actually includes the environment. And at this point, the boundary between the organism and its environment becomes impossible to specify. In other words, not only did the organism and its environment define and co-construct each other, they interpenetrate. And that means we have to study them differently than we've been doing. So, you know, one of the observations that Lewontin made was that in a conventional view, the organism is passive in 2 ways. Its features are dictated by its genes, and its evolutionary fate is decided by the external environmental filter of natural selection. And I think an eco-devo approach and the kind of data we get point to a different view of the organism as an active participant, which I think Ali referred to. The organism has agency. The developing organism integrates genetic and environmental information to shape its phenotype, which in turn determines how the organism experiences and changes its environment. So it has agency in its development, and by modulating its selective conditions and its expressed genetic variation, it also participates in its own further evolution. So here we are back at the dialectical biologist. The organism and the environment, one acquires its properties from its relation to the other. The properties of both evolve as a consequence of their interpenetration. What a beautiful quote, huh? The more we learn about how the organism and its environment shape and interpenetrate each other, the less meaningful it seems to restrict our focus to allelic variation in isolation. And the more important it becomes to bring the environmental context within and across generations into our experiments. Once biologists open the boundary between the organism and its environment, as Lewontin so eloquently argued, we will have to find ways to focus in both development and evolution on the organism-environment relationship. And with that, I thank my wonderful junior collaborators in my lab, and I thank you for your attention.
Thank you very much, Sonia. Gracias a la doctora Sonia Sultan por su presentación y por recordarnos este largo arco entre aquel planteamiento de Lewontin y Lewins en torno al organismo como sujeto y objeto de la evolución y lo que esto significa en términos de datos obtenidos. Thank you very much for reminding us the data arch that links the affirmation of Lewins and Lewontin of the organism being subject and object of evolution, and what is currently being obtained from the experimental data in several systems. So, pues les agradecemos a los ponentes.